Yeah, so for those who don't know me, I'm an assistant professor of soil health based at the Northwestern Washington Research and Extension Center in Mount Vernon. And I've been with WSU about almost 6 years and have been working on this project almost that long. Um, and so I'll tell you, what we did, kind of some of the questions we had going in, and some of the questions we still have, and things we're working on. Many of you may be aware of this project, so hopefully this will be a good update. Excuse me. First, I want to acknowledge all of the folks who have been working on this project, some of whom are here, I think. But this is a project that I am co-leading with my collaborator, Danny Gilardi, who's a senior soil scientist with WSDA. And it started really as a specialty crop block grant in 2020. We were focused on particular specialty crops with the specialty crop block grant. And then with the start of the soil health initiative, it has really expanded. WSDA has kind of taken over a lot of the coordination with it. And we've kind of really tried to expand it to lots of other cropping systems and land use types to really understand what soil health indicators look like across all of these places. And I'll talk more about that. But I also want to acknowledge JD Ryan, who's our data scientist managing all our database, Luis Reyes Rojas, who's a postdoc with us, who's doing a lot of the nitty-gritty data analysis, Leslie Michael, Kobe Sarpong, Molly McElwam, and many of our conservation district partners who did a lot of the sampling for us as we got into this kind of expansion period. So the reason we started this project was kind of a question that came up around, I mean, it's been kind of a question for a while, but just how healthy are the soils that we have in Washington, and how do we define that for Washington state and for the Western US, and how do we define that for different systems? We have obviously very productive systems in Washington and a huge diversity of systems, but the goals of those systems vary, and so how we think about what a healthy soil is in these different systems may vary as well. And also the challenges or limitations, the natural resource concerns may also vary across the state. In some places, erosion might be the biggest soil health challenge. In other places, it might be disease pressure or high or low pH or salinity. So this is something we kind of set out to think about across Washington systems. And these are just some photos I took. Obviously, we have very healthy soils in some cases, but these are some, challenges that I observed as we went around the state and sampled in different places. Some of this impetus also came out of the Washington Soil Health Initiative Roadmap, which I didn't put a link to, but if you're not aware of, I think Molly mentioned it last month. But a couple of the cross-cutting priorities from the roadmap that was done in 2021 excuse me, were to get a better understanding of soil health measurements and metrics and have them be more standardized across labs, and kind of understand the ways that these kind of more high throughput, inexpensive measurements can help us understand soil health, and to just get a better understanding of soil health specific to different production systems. So some of you may be familiar, but these kind of soil health indicators have been used across many systems across the country and across the world to kind of gauge soil health. And there's chemical indicators, that many of which are common in soil fertility testing. There's physical indicators that are being used more, things like aggregate stability, water infiltration rate, compaction levels or bulk density, water holding capacity. Some of these can be done in the field, or soils can be brought into the lab. And then more and more with soil health, there's more biological indicators that are being used, and there's a wide range of these, some of which are very high throughput, inexpensive, things like respiration, where we can take soil that's dry and kind of re-wet it and see how much CO2 comes off and kind of use that as a proxy for microbial activity. So it's kind of like a chemical proxy for biological activity. And then there's also kind of more intensive biological measurements that are done looking at fauna or microbes, looking at those communities. So, and the question is, how do these differ across regions and how can we tie them to important functions in each of these systems? So there's been a lot of soil health work done with these indicators. A lot of it has been done in other regions of the US. So in the Northeast US, there's been a lot of work done by Cornell and others, and kind of trying to understand the typical ranges we would see for these different indicators so that farmers can send in a soil sample and get a soil health test and kind of understand compared to other soils that are similar in the region, where they fall along a scoring curve. And these are developed based on kind of these distributions of a particular indicator across a region. There's a lot of thoughts about, is this scoring curve really the best way to gauge the kind of your soil health level? And there's a lot of discussion around that currently. But the real thing is that a lot of this information has been done in other places. And so, in a place like Washington, where we have huge diversity of climate and soil types, and in some cases, soils that just have lower capacity to, for example, build organic matter, we can't compare those to soils that are in a very different climate and region. So we really need to understand more of what's going on in our state. And so that's kind of the impetus for this project. So the State of the Soils assessment, our first objective was to understand how climate, crop, and management impact these indicators. And thus far, we've kind of wrapped up our sampling actually now this year. And we have over 1,200 samples that have been taken across the state. So this map is a little bit out of date, but you can go to it and it's very interactive. If you haven't played around with it, you can kind of click in and out of different cropping systems and different counties and just kind of see where samples have been taken. Our second objective was to kind of help producers and ag professionals assess soil health, so making some of the information about the indicators a little bit more interpretable, even though there's a lot of questions still in the soil health science space about how these indicators tie to function or important outcomes. And I saw some great questions in the chat, just about interpretation. And I think those are really, really important. But that was one goal, was just to kind of try to get some of this information a little more accessible and interpretable. And then to develop crop and region specific decision support tools so that we do have a better understanding of, what are typical soil health indicator values for different regions. And again, these are just some examples of scoring curves that have been developed in other places. So, excuse me. Just starting to look at the data, our samples range across a huge texture gradient, which is great. We don't have that many fine textured samples in this, clayier side, but we have a big range in soil texture. So we're able to look at the effect that soil texture or sand, silt, and clay content has on some indicators, and then kind of adjust for that when we're doing some other comparisons, looking at, for example, management. And this is just showing the distribution of different indicators by textural class, so coarse, medium, and fine. And these are, for water holding capacity, this is what we would expect, that these coarse soils over here in green have lower water holding capacity. And as you get more fine textured into the clays, there's higher water holding capacity. So, just kind of starting to explore the data, we see what we would expect in terms of texture. Interestingly, with total carbon, which is definitely, thought to be affected by texture or is affected by texture, we don't see a huge difference in terms of median carbon content across our textural classes. Those we see, there are certainly some finer textured soils that have higher carbon. So this is just, as we're starting to explore the data, this is a typical way that soils are kind of categorized by coarse, medium, and fine. Is this the best way to categorize our soils? That's something we're still exploring. I mentioned the scoring curves. This is just one parameter, permanganate oxidizable carbon, and this is just showing an example of why it's important to kind of calibrate these for Washington. So this is the Cornell scoring curve for permanganate oxidizable carbon, and you can see that the top 20% of their distribution is between 600 and 1200 parts per million of POC. But when we look at our dryland systems, we have 0% in that range. So again, if a farmer from Eastern Washington dryland sent a sample into Cornell, if it were being based on a Cornell scoring curve, they would get a failing score. But that's just because it's, inherent properties are kind of driving a lot of this. So again, it's important to kind of calibrate and understand what are the typical values we expect for our area. So Luis Reyes Rojas is I mentioned he's a postdoc, and he's really trying to understand the role of inherent soil and climatic properties on potential soil health capacity. And there's other groups working on this in other parts of the world, really thinking about kind of the effect of climatic properties like mean annual temperature and precipitation, and then inherent soil properties like mineralogy or texture that I was talking about. And thinking about how we kind of classify those into soil health zones. And this is something that Luis is working on for Washington. So, do we want to classify this by textural class, or the Soil Health Institute is also, they've developed these soil health management groups that are based on drainage classes and textural classes kind of combined. And so Luis he has a background in petametrics, so digital soil mapping and using mathematical modeling to kind of predict soil properties. And so we're going to be working on kind of defining soil health management zones in the state so that we can compare kind of apples to apples within these zones, or soils to soils within these zones. And then on top of that, we can ask, how does management affect soil properties in those zones? So we can look at that as well. And then, we're asking the question, what is the best way to categorize these zones? Some people do it by these major land use resource areas, but we think that, and these are just showing some distributions by MLRA, and it's maybe not very meaningful, but I think there's better ways that we can categorize. So I mentioned we looked at the effective management. So we did grower interviews and surveys with all of the people whose fields we sampled, and many of the conservation districts were involved in helping us get all that information. So thank you if you are one of those people. We have information on rotation history, amendments that have been applied in the last 5 years, tillage practices, irrigation, soil challenges, and we also did get yield information. In some cases, though, with the range of crops, that's been challenging to parse out. But we've been working on kind of cleaning up all of this management data and using it to kind of create these indices where we can look at, the effects of different practices. So, for example, we're looking at how are soil health indicators affected by soil disturbance. So in some cases, we've been able to develop this tillage intensity rating for each field based on this 5-year rotation history. And we're combining information based on, what implements are used, how many passes, the depth that those went to, and the percent of the area tilled, to then create one quantitative variable that then we can look at effects of. We've also been looking at cover cropping, mostly treating this as a binary variable, like has there been cover cropping in the last 5 years, yes or no? We've looked at crop diversity in rotation, so calculating a crop diversity index over the 5-year rotation. And obviously, this is something that needs to be very system specific, because certain systems, the capacity to build in other other crops is going to be more limited than others. And so we need to really think about this within a particular context and not compare, a dryland wheat canola system to a maybe irrigated vegetable system, but compare within those, within a particular cropping system, are there farms that are increasing their diversity and what impact is that having on soil health? And then we've looked at, organic matter amendments. Has there been compost, manure, humic acids, biochar, et cetera? We haven't been able to dig into, application rates with this. It's mostly been, again, a binary variable of yes or no. So just starting to look at some of that data in potato systems and understanding the effective management. We've looked at this tillage intensity rating. This is work that my PhD student Kobe Sarpong is working on, and looking at in the Columbia Basin, which is in red, and in Skagit Valley potatoes, which is in blue. How is this tillage intensity related to compaction or bulk density? And you can see that the statistical results here, not a great relationship in either of them, but we do see that and in this statistical model, we've accounted for things like clay content and mean annual precipitation. So we're kind of trying to pull out the inherent effects of soil in this. And we do see that in fields with higher tillage intensity in the Columbia Basin, we do tend to see somewhat higher bulk density. There is a significant relationship, though it's not very significant. But across this huge array of huge variability in fields, we do see some weak trend. So that might be something to think about there with what are the ways that we can minimize soil disturbance in potato systems. And again, this is looking at a 5-year history. So it's not just how much disturbance was there in the potato year, but how much disturbance was there within a 5-year period in a potato system. We've also looked at the effects of tillage intensity in wheat systems. And with this, that we weren't able to do the full tillage intensity rating because we didn't have information on depth and number of passes and et cetera. But what we did was we took the operational stir value for the primary tillage implement, and we categorized those into low, medium, and high, or minimum, medium, and high intensity. And then we were able to kind of look at the response of different variables. So here we have high tillage intensity, medium, and low. And we actually, we don't see any significant relationship between these categories and these two parameters I'm showing here. So pH, while we do see maybe slightly lower mean and median soil pH in the minimum till, it's not a significant difference. That slight trend might maybe due to acidification that sometimes does happen in no-till systems, where you're not getting the mixing, the same mixing, and therefore there's more issues with acidification. And for total carbon, again, we're not seeing a significant relationship. We have a few more sites here that are higher carbon in the minimum till, but it's not a significant relationship. And again, this is across all of the dryland wheat in Eastern Washington. So it's capturing a pretty big area. And again, we controlled for things like clay content and mean annual precipitation and temperature. Effects of compost application in vineyards. This was something my student Molly McElmom looked at, and we looked at carbon, permanganate oxidizable carbon, mineralizable nitrogen, and electrical conductivity. And the only one where we saw a relationship was that there was slightly higher permanganate oxidizable carbon in fields that had compost in these wine grape vineyards. We were glad to not see an effect on EC because that can be a concern with compost application in vineyards. But again, we're not seeing a strong relationship there. So this is just some examples of ways that we're trying to parse out the management data, and now we're looking at kind of the whole management. We just got our last management surveys for 2024. So the ones I've been showing are kind of cropping system specific, and now we're kind of looking at the management data across the whole state, and we'll be able to do kind of more robust analyses with now over 1,200 data points. Another thing we've done, which some of you may be aware of, is we've created these interactive reports for farmers where they can see where their fields fall for a particular parameter compared to all of the fields we've sampled and those in the same crop and county. And we've tried to provide just some interpretation of each of these measurements and linking them to resources where they can learn more about them. And this past March, we launched this R package where others can go in and import data and create these interactive reports and kind of customize them for their farmers. And we just released a Spanish translated version as well. So if you haven't played around with this and it's something that you're interested in, I encourage you to do so. And if you have any trouble with it, we're definitely looking for feedback as well of ways that we can improve it. Another thing we've been working on is method optimization. So in all of this, we realized that there were some inconsistencies in some of the methods for some of these newer protocols. Like this is looking at ACE soil protein, which is a measurement of organic nitrogen. And we found that different labs were actually interpreting the protocols differently. And so we ended up having very different results between different labs. So that's one of our goals too, is again, that standardization of some of these methods. And now we have the labs on the same page with how they're doing it, whether they're doing, 8 mm sieve versus 2 mm sieve soil, and the different the reagent soil ratios that they're using. So this is also important work that we didn't really anticipate having to do, but it's good that it's happening. Okay, so some next steps. As I mentioned, Luis is on, and he's really he's going to be doing some soil carbon prediction maps, for example, looking at how things like uh, inherent soil properties and climate, and then layering on management are influencing soil carbon, for example, across the state. We're going to be doing clustering analysis to kind of create these soil health zones within the state based on the data that we have, and then looking at the impacts of management as well. Our database will be publicly available, and we're working on data exploration tools for folks. And then, something that I think needs to continue to happen with soil health in general is, it's all well and good to know what your indicator values are and say, okay, great, maybe I have a higher POC value than my neighbor, or I have a higher POC value than I did 5 years ago. We have assumptions that a lot of these, increases are good for a lot of these indicators, but we still need to work to tie these to outcomes, and those outcomes might be different in different systems. And so, and there could be a variety of outcomes that are important. So, reduced input use, for example, increased crop yield or maintenance of crop yield, increased farm profit, and then environmental components like air quality, water quality, decreased erosion. Some of these are very difficult to measure, especially across the scale that we were taking samples. And so we can pair this with more experimental work, more controlled work that's going on across the state to really understand how these indicators are tied to outcomes. And this is work that I'm doing with other projects where we have, a lot of yield data, for example, and we can actually look at the effect of an indicator on a yield outcome or on a crop quality outcome. As I mentioned, we collected yield data if we could, but it's hard to parse out because there's so many different crops and different cultivars or varieties that are used. And so how do we kind of normalize that and think about it within a system? And we've been able to do that within some systems, but we don't have enough data for other systems. But as we think about this, these soil health interpretations, this is maybe it's a point of discussion that we can have today, but just how can we better tie these to outcomes so that we can, actually know that, okay, an increase in this indicator is actually having a benefit for my farm or for the surrounding environment. So with that, I'm happy to open it up for questions and discussion. And as you can see, this is still a work in progress, even though we've been working on it for 5 years. But we have a very rich data set, and there's a lot we are working on with it, and probably other things we could do with it. And happy to get ideas and feedback.